Okay, Bismillah ar-Rahman ar-Rahim. Assalamu alaikum jami'an. This is my lecture on this section about digestion and absorption of proteins. My name is Yasmin Abusamra. I'm a professor of biochemistry and medical genetics at Sonoran University in Arizona. And I'm also the Division Director of Human Sciences and Department Chair of Basic Medical Sciences. I work in Arizona, but I live in Texas, and I work fully remotely. So the experience with the SVMA is a challenge to me because I am experienced, of course, with the online teaching, but the challenge here is that I have students from all over the world, from many different countries. So I hope that you have stable internet, internet connections and electricity, so we can together go through this topic. Okay, so now as you can see that I will be talking about proteins, and proteins is one of the macronutrients that when we teach biochemistry, we focus on 4 macronutrients. We call them macro because we need them and we use them in bulky amounts. So proteins is the first, or normally when we teach protein, it comes the third on the list. So we start with carbohydrates, then we go to lipids, proteins, and finally nucleic acids. So today this is the first lecture in proteins and absorption, digestion and absorption of proteins. So in this lecture, I'm going to go over the digestive enzymes. And it's very important when we study biochemistry, we focus on enzymes. And of course, later on, we'll come to the regulation by hormones. But I always tell my students, if you want like to be a good biochemist, you have to know your enzymes, okay? So every time or any time you study any topic in biochemistry, make sure to know the names and functions of the enzymes, and if we have deficiency on these enzymes, what will be the consequences, okay? If you know these items about your enzymes, definitely you know your biochemistry topics. Okay, and then after that, I'm going to talk about, after we digest the proteins that we take from the diet, I'm looking at the absorption, how we absorb those molecules and how we utilize them. I mean, like, you know, when you eat a piece of meat, for example, so this is proteins, right? But you cannot use the whole protein inside your cells. You cannot take it from your mouth directly to your cells. So you have to digest it, you have to absorb it, and then send it to your cells where you can utilize it and you can benefit from it. After that, we're going to talk about the defects in amino acid absorption. If for some reason a person or an individual are unable to absorb those amino acids, what will happen in their systems? And then I'm going to talk about protein quality and turnover. And this section will tell you about the type of proteins that you are eating, not only the quantity, but also the quality of proteins. So is it differ between, what's the difference between like consuming plant proteins or animal proteins? And about the turnover is how we utilize them, what we lose, what we gain, what we store, or of course we don't store proteins, but what do we keep them as proteins for certain times and how do we degrade them? And the recommended intake, that's how much do we need from those proteins? And finally, what will happen if we have protein deficiency? So we're going to start from your table, from the food that you take, until we get to the point what will be the consequences of protein deficiency. Okay, so when we talk about protein digestion, we start in the digestive tract and we look at which enzymes are involved. And I told you, in biochemistry, we're going to focus all the time on our enzymes. So the digestion normally will start in your mouth, and we call it mechanical digestion, and this is for all types of food, because if you take a bulky protein from your food, okay, you have to chew it, right? So this is what's called mechanical digestion. That means you are breaking it down into smaller molecules until we get to the stomach, and there we start the actual digestion of proteins. And in the stomach, we should see two things. The first one is the hydrochloric acid, and the acidity or any change in pH will affect the protein structure. So hydrochloric acid will start to denature the protein, and this is part of the breaking up the breaking down the bonding between different amino acids, okay, or trying to denature the protein, okay. And after that, we have also pepsin. Pepsin is found in the stomach as pepsinogen, and this is the proenzyme, so or inactive enzyme found in the stomach. Once you get your proteins to your stomach, then pepsinogen should be stimulated and activated and becomes a pepsin. Okay. So all this will happen in our stomach, and the end product, instead of having a whole protein or big bulky proteins, now will start to degrade it down. Okay, and next we have to move the digested portion of the protein to the intestine, and there we're going to see some other peptidases. We call them peptidases, so it could be either endopeptidases or exopeptidases that act on the proteins either from the ends, from the middle, okay, degrading it from the middle, or start to remove amino acids from the ends, which is exopeptidases. So after that, we're going to have our final products, which could be either just single amino acids or dipeptides or tripeptides, and those could be absorbed easily when we compare them to the starting point here, which is the bulky protein. Okay, so besides these enzymes, once we have the food, then we will see that there is an secretion of certain hormones. Okay, so we're going to talk about two of them here. The first one is called cholecystokinin or CCK, and the other one is secretin. These two hormones will regulate secretion of some we call them fluids that will contain like bicarbonate, which will change the pH of the intestine and make it basic, which is very good media. Basic media is a good media for the enzymes that act in the intestine, and also the CCK will help us to stimulate and activate another enzyme called trypsin. Okay, so trypsin is one of the very important enzymes in protein digestion. And before we have CCK and before we have the protein from the diet, trypsin is found as trypsinogen, which is the not active form of trypsin. Okay, so now we move to this diagram, which is really very helpful to show you exactly what will happen. So first of all, we see that gastric cells will release the hormone gastrin, and this will start to release gastric juice in the stomach. That includes hydrochloric acid and pepsin. Okay, so this is what happens in the stomach. After that, we're going to see CCK and secretin are secreted and help to stimulate the release of bicarbonate, and also some other pancreatic enzymes will be activated because now they have the basic media where they can act. And finally, we see the products, which are, as I mentioned, either amino acids, dipeptides, or tripeptides. Also, I really like this table because it will show you the inactive forms of the enzymes and the active forms of the enzymes in this column, and also which stimulators, which molecules will activate the enzymes, the site where the enzymes could be active or acting, and which substrate, which types of amino acids the enzymes can break down or degrade, and finally it will show you the end products. So let me start with the stomach. This is the first place we should be able to actually digest our proteins. And in the stomach, we should have pepsin. So before the secretion of hydrochloric acid, the pepsin should be found as zymogen or inactive enzyme called pepsinogen. Okay. Now we have our proteins, gastric juice are secreted, including hydrochloric acid. Then pepsinogen will be converted to pepsin. Now it's active. Active where? In the stomach. And what will happen after we have pepsin? The pepsin will act on most amino acids, including aromatic, dicarboxylic, leucine, methionine. It has variety of amino acids that can act on and degrade, because the main point of digestion is getting smaller molecules. And this is true because the end product here is are peptides, which are smaller molecules rather than the full proteins. Next, we're going to see trypsin, found as trypsinogen, act in the intestine, and all the rest will be in the intestine, small intestine, and it will act into on basic amino acids. And again, we're going to get more smaller peptides and some of the free amino acids. Now we're going to move to chymotrypsin and finally the carboxy peptidases. Once we get to this point, we are going to see more of the free amino acids. Those are enzymes that are activated by trypsin and act on either the C-terminal or N-terminal of the amino acids. That means from the end until we get free amino acids. So this table will show you different enzymes, and as you can see, we're going to see trypsin mentioned few times. So either it will act on the protein by itself, or it should be able to stimulate some other very important enzymes to act on the protein. So that's why there is always an argument about trypsin being the most important enzyme in protein digestion. Okay, any questions so far? Do you have questions? Please unmute yourself and ask your question if you have one.
No.
Sure. Can I hear from other students, please? Any question about digestion? So, in summary, you take a bulky protein, try to digest it into smaller molecules, because this is the way that you can absorb it and you utilize it. Okay, so I'm always telling my students interesting story about collagen. Okay, so because many manufacturers are trying to sell the collagen as an amazing product for skincare, for example, but there are two things about collagen. Number one, if you apply it topically, like cream, collagen cannot be absorbed unless if it's synthetic collagen, which is smaller, because collagen is a very, very big molecule, so it cannot penetrate through your skin. Okay, so this is number one. The second thing, if you take protein, your collagen is a protein. So if you take it as pills or part of your food, it should be degraded to smaller amino acids or tripeptides, dipeptides, or free amino acids. After that, you will absorb it, and then it has to follow the synthetic pathways in your skin to become collagen one more time. Okay? But it doesn't mean that if you eat other proteins that contains the same amino acids in collagen, it doesn't mean that you cannot synthesize collagen in your skin from the availability of other amino acids. So whatever you take, any type of proteins, you have to digest it first. There is no way that you take your collagen by mouth and then it will go directly to your skin. Okay, so we have to be very careful about what's telling our patients in the future, because we have to build a trust. Okay, and yes, it's good to take collagen. Okay, because by the end of the day, it should be degraded into amino acids, and this is what you need to synthesize a healthy protein for your skin. But it doesn't mean that if I take some other proteins, it should be able to do the same thing. Okay, this is number one. Number two, normally when we take proteins, we don't take them as free amino acids. Most of the time, we take them as complete proteins, and then we have to degrade them with just very few exceptions. There are some amino acids powders. People are taking them when they are building up their muscles. So these are the things that might be easy for digestion and absorption. But other than that, normally we take complete proteins and start to degrade them. So after we end or complete our digestion, now we do have amino acids, dipeptides and tripeptides ready for the absorption. So this is the second part. Okay, until then we are in the GIT tract. Okay, like mouth, um, stomach, intestine, we are still there until we complete the absorption. And after we absorb the amino acids or dipeptides, they will go directly to the circulation, to the blood. And from there, we can send them to our cells, where we can use them to either synthesize proteins, or we have some other categories, like we call them specialized products like purines or heme, or some other molecules that are synthesized from amino acids other than proteins. Okay, so how do we absorb those amino acids? Okay, if you just consider them free amino acids, how do we absorb them? And in general, the absorption mechanisms could be either by diffusion or facilitated diffusion or active transport. Diffusion that goes directly through the cell membrane. Facilitated diffusion normally requires a transporter protein that's found in the cell membrane, or active transport where we need a transporter plus energy. But for protein absorption, normally we use facilitated diffusion. So when we absorb our amino acids, they have to cross two borders or two membranes in the the small intestine. The first one is called brush border membrane. The other one is called basolateral membrane. So this is the brush border, and this is the basolateral, and this is the lumen for the intestine, so inside the intestine, and this is the enterocyte, which is the intestinal wall. So we want to take it from the intestine to the blood. So it has to cross these two membranes, and this is a transporter, okay, for the peptides, so they can enter the cells for the intestine. And when it gets here, it comes with protons. Okay, but because the proton has a charge, we have to get it back again outside, and then sodium will enter. And on the other side, sodium will leave, potassium will enter, and so on and so forth. All this mechanism is just to help the peptides or amino acids to get into the sites or intestinal walls here. After we get them here, now we need to move them to the blood, because the blood is the carrier for the amino acids to send them to your different cells. Let's assume there is an X cell here or Y cell here, need to make some proteins to do, for example, glycolysis or fatty acid synthesis or whatever, any mechanism that needs a protein or an enzyme. Then the amino acids will go to the blood, and from there to the actual cell. Okay, so after we get them to the blood, now we need to know how those amino acids can enter the cell. We have different mechanisms, but this is one of them. Okay, many mechanisms by which we can get the amino acids inside the cell. This is the major form, and as you can see here, it's called sodium, sodium has to bind to the transporter. So this is the cell membrane, and as you can see that it has lipid bilayer. Okay, amino acids in general, they are hydrophilic in nature, so they need transporter to cross the lipid bilayer over here. So sodium is a helper in this case. So the sodium will bind to the transporter. What will happen? The transporter now is ready and stimulated to bind to the amino acid. Now sodium amino acids are bound to the transporter, and they will make a conformational change. The transporter change their structure in a way that allow both sodium and amino acid to enter the cell. Once we have the amino acid inside the cell, now the cell will decide what to do with it. Okay, if it needs an enzyme, it can use those amino acids to make the enzyme. If it would like to make any kind of maybe neurotransmitter or anything, okay, any molecule that requires this nitrogen, then it will use that amino acid to synthesize it. But the sodium has to leave back through sodium-potassium channel and wait for another amino acid to help it to go inside the cell. So now the story is completed. We know that we can take protein from the diet, we can digest it, we can absorb it, and we can send it to the cell. And this is our goal. We need our amino acids to get here. And this is just the cell, not a specific cell. Okay, the cell that requires amino acids to synthesize proteins. Any questions so far about digestion and absorption?
No.
Okay, perfect. Okay, so next we need to know if we are unable to do this process. Because when we study biochemistry, we need to understand the normal mechanism and also the abnormal mechanism and what will be the consequences. So now we need to look at the defects in amino acid absorption. Okay, we have a good example, and just remember, I cannot teach you everything, right? And these are just examples of what will happen if we are unable to transport specific amino acids which has neutral R group, or they are neutral amino acids. Okay, so neutral amino acids, and a good example here is tryptophan. Tryptophan, if you remember the classification of amino acids, we have acidic amino acids, we have basic amino acids, we have amino acids that has benzene rings, we do have amino acids that are neutral, they don't have any charges, polar, non-polar. We have different types of amino acids, and based on that, the transporters will be different. Okay, so the transporter that are transporting neutral amino acids are different from the transporters that are transporting amino acids that have charges. Okay, so those patients with heart nub disorder, they have defective transporters for neural amino acids, and specifically tryptophan. So what does that mean? They don't have transporters. That means their cells cannot accept tryptophan. Okay, this will create two conditions. Number one, in order to make any protein or any enzyme in your cells, you have to have 20 amino acids. One of them is tryptophan. So if you cannot get it in your cell, that means that cell is unable or not capable of synthesizing proteins. This is number one. Number two, tryptophan is used to form niacin. Niacin is a vitamin that if we don't have it, it will create a condition, and it's called butterfly, okay, skin rashes. So the shape of the rash here, it looks like a butterfly, and that's an indication of low niacin. And we have low niacin because we don't have tryptophan that's used to synthesize niacin. This could be treated by, increase the amount of niacin from the diet, because niacin is vitamin B3. So it's treatable. But heart block disease or disorder is not treatable because there is a defect in the transporters of tryptophan. Even if you increase the amount of tryptophan from the diet, still those patients are unable to accept it and take it inside the cell. Of course, there are some other ways to treat those kind of symptoms. Now we need to move to the quality of proteins, turnover, recommended intake, and deficiency. We're going to see each case by itself. So the protein quality. Okay, so normally the animal proteins are sufficient. They have all the 20 amino acids, and you should be able, if you're eating animal proteins, to get all the amino acids that's required for protein synthesis. So we don't have any issues with animal proteins. With plant proteins, some of them have incomplete proteins, okay? Like in some other nations where they consume certain types of food that deficient in lysine. This is very common in Central Africa. So the protein that they are taking from their food, and if I remember correctly, the food called cassava, I think, but I'm not positive. But it doesn't have lysine in it. And then when people consuming that protein, it should have less quality. And when those individuals or those people, when they're trying to synthesize their protein, there are one missing, which is lysine, and there is no way to synthesize all types of proteins with a missing protein. So we have missing amino acid lysine. So two things you need to consider here. Number one, the quantity of proteins. You need to get enough proteins. The second thing is the quality of protein, and you could do it by supplementation, and you have to make sure that the protein also is digestible. Okay, so we need to make sure that we are getting the correct amount and also the quality, specifically the 10 essential amino acids that we cannot synthesize in our cells. Okay, so every day we have a protein turnover. So about 1 to 2% of the protein each day that are turned over, and we don't store the proteins. Okay, so we synthesize proteins, use them, and then we degrade them, and we degrade some of them completely, even the carbon skeleton, and get rid of the ammonia. So how do we do that? We do it by certain enzymes, and also they are protein in nature. They are called proteases. So let's assume that you did one of the pathways and you're done, and you don't need to do it again recently or soon. Then those proteases will act on those enzymes and degrade them and get them to the amino acids level, and those amino acids are sent to the circulation one more time, and they stay there circulating until there is a call from one of the cells, hey, I need to synthesize some proteins. Then those amino acids will go there through the transporters, and the cell will use them to satisfy their cellular needs. Okay, so where do we degrade those proteins? The proteins are degraded mainly in the lysosomes and proteasomes. So lysosome degradation, this is a very normal process, and we have lysosomes in all mammals or mammalian cells except the red blood cells. So we don't have them in the red blood cells. So there we in the lysosomes we do have digestive enzymes for proteins, carbohydrates, lipids, and nucleic acids. So everything that we don't use, we send it to our lysosomes, and there they are degraded. And even the degradation or digestion, lysosomal digestion, it doesn't even requires energy. So it's a simple process. There's also a process called autophagy or self-eating. So if there's a cell that's not needed, it will be degraded, and their protein also will be degraded. The proteasomal degradation is a little bit different from the lysosomal degradation. Still, we degrade the proteins here, but in this case, mainly we degrade the abnormal or damaged or denatured or proteins that are found in the wrong place, or also the regulatory proteins. Regulatory proteins should have very short lives. We just need them to regulate some process, and then we have to degrade them. We don't need them anymore. So the proteasomes will degrade them. So how the proteasomes will recognize those proteins, how the proteasome will figure out whether the protein is a normal or damaged protein. So normally the damaged proteins will be bound to a molecule, small molecule called ubiquitin, and the process is called ubiquitination. So if a protein has a ubiquitin that's bound to it, then the proteasome will recognize it as a damaged one, and then it will start to degrade it. So now we need to know how much protein do we need. We have standards here, which is 0.8 gram per kilogram. So if your weight is 70 kilograms, you need to multiply this by 0.8, which is about 56 grams per day. This number varies dramatically, okay? So definitely the amount of proteins that you need, it's definitely different from the amount of proteins that I need, okay? So it depends on the physical activity, health conditions, weight, gender. Activities is the most important thing, okay? So if an athlete is trying to build up muscles, definitely they use more than this amount. And I always give an example of my son, because he's trying to build up muscles and he's using 3 times of this amount. As you can see, it's also different from newborns, children, adults. So there are so many factors affecting the amount of proteins. It's not just the amount that we need to take, but also it has to include all the indispensable or essential amino acids. So this is the most important part of it. It should contain essential amino acids. So some studies shows that there is no negative effect of high proteins, but I always tell my students that even high protein intake is not harmful, okay? Still it will affect the absorption of other molecules. So we have to be careful, we have to be balanced when we are taking our macromolecules, and we also have to be very wise when we advise your patients in the future. Okay, so this is about how much do we need from the proteins. Then now finally, we need to talk about protein deficiency and malnutrition. Okay, so we have few conditions where people could suffer from full malnutrition or just the protein deficiency. So kwashiorkor is a condition where those individuals or those people are able to get enough energy. What do we mean by enough energy? We mean that they should be able to get enough carbohydrates, enough lipids, but insufficient proteins. Okay, so either the quantity or quality, but mostly here the quantity. They don't get access to enough proteins, or the proteins that they are consuming is not complete. So those will develop what's called as kwashiorkor, and it's characterized by edema due to loss of blood proteins. So let me tell you one more time. If you are trying to synthesize your proteins, you need 20 amino acids. If they are taking protein that doesn't contain the 20 amino acids, then this will lower the amount of proteins that they are synthesizing, okay? And including, of course, blood proteins. So what's the correlation between blood proteins and edema? The blood proteins contains, or one of them is called albumin, And the albumin is found in the blood vessels, holding water inside the blood vessels. If we lose those albumins, or if we have low level of albumins, then the water will diffuse from the blood vessels into the space between the vessels and the cells. Okay, so that region should not contain that much of the water. If the water is released in there, it will cause edema, which is like, you're going to see like, big bellies. Okay, and that's because we have this water diffuses out of the blood vessels. In marasmus, which is the second case, normally we see the same symptoms, but in this case it's not just about the amount of protein that's not sufficient or the quality of the protein, but this is about the whole about malnutrition. So this is including even the energy molecules like carbohydrates or lipids, and of course including proteins. So this will result also in edema and also muscle wasting. And here also in the kwashiorkor, if you look at them, they characterized by big belly but also very skinny. Okay, because they are losing their muscle proteins. Here also we see like muscle wasting and edema, and because they need all of the above, like energy molecules plus proteins. So in summary, again, if we look at protein digestion, we need to focus on different things or different points. Number one, you need to understand that when you eat your protein, normally it's not in the simpler form or the building blocks, which are the amino acids. You eat your protein as a complete protein that has either three tertiary structure or even a quaternary structure. So we start with the digestion in the mouth, okay, which is the mechanical digestion, just breaking up some of the bonds, and then go to the stomach, where due to the presence of the food, your stomach will start to release the hydrochloric acid and stimulate or activate pepsin. Okay, both of them will act on the proteins, getting you, instead of having a big protein, now you're going to get smaller peptides. The digestion will be completed into small intestine, where we see that due to the secretion of two hormones, which are secretin and CCK, we're going to see an active environment for intestinal enzymes to act further on those peptides to get us either free amino acids, dipeptides, or tripeptides. Once we complete the digestion, we need to absorb it through different mechanisms and different transporters depending on the structure of the amino acid. And finally, we're going to build up a amino acid pool in the blood or the circulation, where could be circulated and taken up by certain cells. Once we get there, the cell will use it to synthesize proteins or some other non-protein compounds. And we talked about the abnormalities when we have defects in the transporter or transporting mechanism of any amino acid. And the good example for that was tryptophan and heart nerve disease. And we also talked about how much proteins that we need per day. The quality of protein, it should contain all the 20 amino acids, and specifically the essential amino acids. And we talked about the turnover of the proteins, that we really don't store the protein. We use it, we degrade it, we lose some of them, and we return the rest. And also the protein deficiency, kwashiorkor and marasmus are good example for that. Any question?
No, everything is clear.
Is heart congenital disease or not?
Okay, I see that we have, I do have 12 attendees here, including myself. Okay, so let me just have a quick feedback. Um, any question about the topic itself? عندكم أي سؤال عن المعلومة نفسها؟ ال contents, protein digestion and absorption.
No, it's pretty easy.
Okay, perfect. And okay, so as biochemists and future physicians, did you get the point here? When you study such a topic, do you know where to focus? What do you need to learn? What should be the outcome? What will be the take home message for you as a future physician and as a biochemist for now? What's the important point here when we study such a topic?
I think we focus on
We have to know the mechanisms and the enzymes used to digest everything.
Okay. One last thing. Yes. What else? Okay. So you need to know abnormalities. Okay. So this is different from people who are studying structural biochemistry. Okay. Or just pure biochemistry. They will focus on the details of the structure, the mechanism, and all related influences and molecules that will affect the pathway itself. But for you as physicians, you really need to understand the abnormalities, the normal pathway and also the abnormal pathway, because this when the patients come to you, they will not come with the normal pathways, right? They will come to see you because they have some issues, and you need to rely to decide which way to go on treating them. Okay, any other questions, please? Are you still recording or recording is stopped? Well,
أيوة يا دكتور سجل في المحاضرة.
Okay, thank you. Okay, well, thank you so much. You have about 9 minutes to go to your next class if you have one. Good luck with your journey with us at SVMA and see you next week.
شكرا يا دكتورة. بس الPDF حينزل ولا حترسليه لنا؟
أنا أصلا نزلته.
تمام.
إذا ما لقينه
شكرا يا دكتورة.
contact me. Thank you so much.
Okay.
Good luck to all my students. Bye bye. See you next week. Maybe not. I'm not sure. Okay.
Okay, thank you.
You're welcome to the end. Bye.